Hi everyone, this is the 14th lecture for your chapter Cell, and with today's lecture we will be finishing the whole chapter. Okay, so let us begin with the first organelle for today, which is ribosomes. Ribosomes are naked in the sense that they do not have a membrane. Okay, so these are one of those organelles which do not have a membrane. Fine, so ribosomes are naked. Ribonucleoprotein, here the name ribonucleo, because it has RNA. If you know the full form of RNA, it is ribonucleic acid, right? So because it has RNA, it is called ribonucleo, and this RNA is found with protein. So it is a ribonucleoprotein particle. Clear? And it is a site for protein synthesis. That is its function. What does the ribosome do? It does the function of protein synthesis. It is made up of two unequal parts. Unequal because one of them is larger, which is called large subunit, as you can see here. Okay, and it is more like dome shaped. You can see, right? And the other one is the smaller subunit, which is ellipsoid. Okay, ellipse shaped, which is like oval in shape. Okay, now to combine these two, okay, to make sure that large and small subunit they can come together, you require magnesium ions. So if there is less amount of magnesium ions, the two parts of a ribosome will not come together. Okay, and if there is only enough or more than enough amount of magnesium ions, will the ribosome units be able to come together? Fine. Then when we talk about the numbers, okay, how many ribosomes are present in a cell, they can occur in a large number. Just for an example, in single cell of E. coli, Escherichia coli, which is a prokaryote, okay, it is a gram-negative bacteria. So in one cell of E. coli, it has about 20,000 to 30,000 ribosomes. Okay, so if you just consider the number of all different organelles, this organelle is present in the highest number. It is 20,000 to 30,000 in bacteria, okay, in E. coli. In eukaryotes, this number is several times more. Fine. Now they may occur single, okay, as we saw in the previous slide, the ribosome may occur singly, in which case it is called a monosome. Mono means one, okay, and some means body, single body or single ribosome. Or they may occur in helical groups over an mRNA. So you can see here in the diagram, this blue colored line that you're seeing is your mRNA, messenger RNA. RNA is single stranded, okay? It only has one strand, and this messenger RNA, it contains the code, it contains the message, the instruction to make proteins. Fine. So here you have one strand of RNA, mRNA, and on it you can see a lot of ribosomes are arranged. Okay, you can find a lot of ribosomes arranged on a single strand of mRNA. These structures are called polyribosomes or polysomes. Both of the things mean the same. Okay, you can either call them polyribosome or you can call them polysome. This is mostly found in prokaryotes, and the reason for this kind of structure is so that from a same transcript, okay, from one mRNA, when you require a lot of protein, so a lot of ribosomes will come and attach on this mRNA, and each ribosome will be synthesizing the same protein, the same polypeptide. Okay, so from one transcript, from one mRNA, you can get multiple copies of the proteins. When the cell is actively synthesizing protein, or it actively requires protein, at that time you find polyribosomes. They occur in all living cells with the exception of mammalian RBC. Okay, ribosomes are present in all living cells, whether it be prokaryote, eukaryote, archaea. The only exception to such cells is RBC, mammalian RBC to be specific. In mammals, okay, in mammals RBC, red blood cells, you do not find ribosomes. by now you must have understood that mammalian RBCs are exception to a lot of organelles. Okay. Now, depending on place of their occurrence, like where they are found, the ribosomes are of two types. They can either be cytoplasmic, meaning they are found in cytoplasm, or they can be organelle type, which means they are found in some organelles. Okay. Now, what organelles have ribosomes? So we have already discussed two. The first one is called plastid, right? Plastid, the ribosomes found in plastid are called plastido-ribosomes. And then we also have discussed mitochondria. So mitochondria also has ribosomes, and these are called mitoribosomes. Fine. So in case of organelle, only two organelles have their own ribosomes. These are plastids and mitochondria. Fine. The rest of the ribosomes are found in cytoplasm. Inside the cytoplasm, they can either be free, okay, which means they are just free living in the cytoplasm, or they can be attached to endoplasmic reticulum. Now, when we talk about the ribosomes, their size, okay, is measured by a unit called Swedberg unit. The Swedberg unit is the sedimentation rate. That is, if you take a test tube and if you have, let us say, these ribosomes, and you apply centrifugal force, which is you circulate it, okay, you make it go round and round. In such case, how long does it take for this subunit or this particle to settle down? That is called sedimentation unit or Swedberg unit. So the cytoplasmic ribosomes of eukaryotes, fine, of eukaryotes are 80S, which means 80 Swedberg unit, and those of prokaryotes are 70S type. The 70S is made up of 50S plus 30S. Okay, we already talked about the ribosome being made up of two subunits. Okay, one is the larger subunit and the other one is the smaller subunit. So from here you can come to know the larger subunit will be 50S and the smaller subunit will be 30S. One thing to remember is here the rules of additions do not directly apply. Okay, 50 plus 30 should give you 80, but they do not make 80. Okay, when the large and the small subunit are bound together and you try to sediment it, it sediments in 70 units. Okay, so 70S is 50 plus 30. Always remember this. Now let us see here what these subunits are made up of. Now I hope you will be able to remember this properly. Sub unit, okay? Unit is any one thing and subunit is the part of that one thing or unit, fine? So this 50S subunit, the larger subunit, it is made up of 5S rRNA. Okay, here S is again Swedberg unit and rRNA is ribosomal RNA. Clear? So this 5S rRNA is a very small RNA, and then you have 23S rRNA. So 5 plus 23S rRNA and some proteins. How many? 34 proteins are found together in this 50S subunit. Clear? So we'll be able to remember this much. The 50S subunit has 5 and 23S rRNA. Then your smaller subunit, which is 30S subunit, has 16S RNA and 21 polypeptides. Polypeptides are nothing but they are protein chains. Fine. So from this you can come to know why we call ribosomes ribonucleoproteins. Okay, because they have this ribosomal RNA as well as they have proteins. So this is how the 50S subunit and 30S subunit are made of, and together they form a 70S prokaryotic ribosome. In eukaryotes, the ribosome is 80S type. Here again, the rules of addition do not apply. 60S will be the larger subunit and 40S will be the smaller subunit. Together, 60S plus 40S gives you 80S. Clear? So the 80S type of ribosomes found in eukaryotes. Now let us consider their large subunit and smaller subunit, what they are made up of. So here the larger subunit is made up of 5s rRNA, 5.8s rRNA, 6.8s rRNA and 28s rRNA. So compared to the 70s RNA, 70s ribosome, you have two more rRNA present in the larger subunit of eukaryotic ribosome. Clear? So this is giving you the 60s subunit along with some proteins. The 40s subunit, which is the smaller subunit, it is made up of 18s rRNA plus more than 33 polypeptides. Okay, and together this 60 plus 40s makes the 80s eukaryotic ribosome. When we talk about the structure of ribosome, again, it looks like this. Okay, this is the larger subunit. It has a thumb-like structure coming out. Can you see? So that is a thumb-like structure like that. Then you have the smaller subunit, and between these two subunits, the larger and the smaller subunit, there is a space where mRNA can fit. Okay, so that will be the mRNA binding site where the mRNA can bind to the ribosome. Clear? Now let us take a cross section of this ribosome mRNA complex to find out how it looks like inside. So inside here you have the small subunit, large subunit, and this is the mRNA. On the inside you have three sites, okay, three different regions: the A site, P site and E site. Clear? A site is for arrival. Okay, here the tRNA, transfer RNA, will come with the amino acid, okay, and they will attach here at the A site. Then you have P site. P site is where the peptide bond is formed, okay, between the two amino acids. In biomolecules you must have studied, in between two amino acids there is a peptide bond. So this peptide bond is made in the P site, and this reaction, okay, is enzymatic reaction, and it is done by the enzyme peptidyl transferase. This peptidyl transferase is the only enzyme which is actually made up of RNA, not proteins. Okay, there are two types of enzymes. One are normal enzymes which are made up of proteins, and others are called ribozymes. Ribozymes are made up of RNA and they have catalytic power. So here the peptidyl transferase protein, okay, peptidyl transferase enzyme is the one which forms peptide bond between the two amino acids. After the tRNA has given its amino acid and there has been a peptide bond formed, that that tRNA will leave the ribosome, okay, and it leaves through the E site. E is for exit. So that tRNA comes to the A site, it binds to the mRNA, then it gives its amino acid to the P site, and then it leaves, okay, the tRNA, it leaves through the E site. So these are the different sites you find in a ribosome. 70s ribosomes of prokaryotes are formed in cytoplasm. Now let us see where they are formed, where their synthesis takes place. So in case of prokaryotes, these 70s ribosomes are made in the cytoplasm. For 80s ribosomes, which are found in the eukaryotes, the single-stranded RNA, wherever you find something like this, ssRNA, it means single-stranded RNA. If it is DS, it means double-stranded RNA. Okay, same thing applies for DNA as well. ssDNA means single-stranded DNA. dsDNA means double-stranded DNA. So in case of 80s ribosome, the single-stranded RNA and proteins are made separately in the nucleolus. Fine. There is a place in the nucleus which looks very dense. Okay, it looks very much colored. So these portions are called nucleolus. When you take a closer look at the nucleolus, it looks something like this. Okay, it is divided into three main parts. The central part where you see the yellow colored band is actually your DNA. Okay, this is the part where you have your DNA. From your DNA, transcription will happen and the rRNA will be synthesized. Okay, transcription is conversion of the message in the DNA into form of RNA, right? So here from this yellow colored DNA of the nucleolus, you get rRNA. Okay, here these purple colored strands or thread like structures we are seeing are all different sizes of RNA. Fine. So here you get the rRNA and then the rRNA plus the different proteins. Okay, they come together and they form the large subunit and the small subunit. They do not become functional inside the nucleolus. So these ribosomes, they do not come together and become functional inside the nucleolus. They are subunits. Okay, the larger subunit, the smaller subunit. They come out of the nucleus into the cytoplasm. Then they come together and become operational. Ribosomes are ribosomes of semi-autonomous organelles, which means ribosomes of plastids, specifically chloroplast and mitochondria. They are also synthesized inside the nucleus. Okay, in nucleus and the specific region called nucleolus. Now, what are the functions of the ribosome? They are known as protein factories of the cell, right? So it is the site for polypeptide or protein synthesis. When we will discuss the biomolecules, I will share the difference between polypeptide and protein. Okay, polypeptide is basically a polymer made up of amino acid. Protein is also the same thing, but it has a 3D or a 4D structure. Fine. So that is the basic difference. It has the enzyme peptidyl transferase, which is a ribozyme. Remember this, and factors to form polypeptide. Free ribosomes synthesize structural and enzymatic protein. Okay, the free ribosomes that you find in the cytoplasm, they synthesize structural proteins. Structural proteins are the proteins which you find on your plasma membrane or in the cytoskeleton, okay, which just provide structure. And enzymatic proteins are the proteins which have catalytic function. So these kind of proteins, structural and enzymatic, are made by free ribosomes. The attached ribosomes, that is the ribosomes found on the endoplasmic reticulum, they synthesize proteins for transport. Okay, as we have already studied the endomembrane system, through this attached ribosomes, the protein, they will be directly synthesized and packaged into the endoplasmic reticulum, from where it will go towards the Golgi body, and from Golgi body or Golgi apparatus it can get finally packaged in form of vesicles and fuse with the cell membrane. Okay, so the proteins which are to be transported or to be secreted are synthesized in the ribosomes which are attached to the endoplasmic reticulum. Then the newly synthesized polypeptide is provided protection from cytoplasmic enzymes by enclosing it in ribosome. So whatever this new polypeptide will be forming, okay, from the ribosome, normally what happens is in your cytoplasm there are certain enzymes like peptidases and some nucleases. They may degrade your protein before it even forms, okay, to protect it so that it can form properly and then release it. So this protection function is also done by the ribosome. Now we move on to the last organelle we have to study, okay, which is the nucleus. It is the most important and integral part of a cell, okay, and the plural of nucleus is nuclei. In Latin, the term nucleus means kernel of a nut. So for example, if this is a peach, then this is called its kernel. Okay, and can you see the resemblance? If you look at this structure of a cell and the purple colored one is your nucleus, it looks very much like kernel of a nut. Okay, so that is why it was given the term nucleus. It was first studied by Antony van Leeuwenhoek. So the same guy, same scientist we have studied, okay, previously in the history session. So that same person, the same scientist, Antony van Leeuwenhoek, he was looking at salmon cells. Okay, salmon is a type of fish. So he was looking at fish cells, and he found, or he observed, these nucleus. However, he just mentioned it. He did not study it in detail. There was another scientist called Robert Brown. This Robert Brown was looking at orchid epidermis. Orchids are type of flowers, and epidermis is the outermost layer of any plant. Okay, so epidermis, this is how it would look like, what he saw. So on epidermis, these things you are seeing are the stomata. Okay, the semilunar shaped guard cells, and the pore in between is the stomata. Fine. So on the epidermis you find stomata. When he was looking at this, he found there are these round small structures, okay, which he called nucleus, and that is how he studied these organelles called nucleus. It is a double membraned organelle, which means like mitochondria and chloroplast, it has two layers. Okay, it has two lipid bilayers. So double membraned organelle that contains the genetic material of eukaryotic cell. Again, nucleus is absent in the prokaryotes. It is only present in the eukaryote. So it is a very good difference, okay, between the prokaryotic cell and the eukaryotic cell. The major difference between the two is presence of nucleus. This nucleus contains all the genetic material, okay, all the inheritable material, the DNA of the cell. Okay, the only exception where there is no no nucleus are the mammalian RBCs. Okay, I keep on emphasizing the term mammalian because in other organism, okay, in lower organisms like frogs or even in birds, you find they have nucleated RBCs, which means they have nucleus in the RBC. Mammalian RBC are the only exception of eukaryotic cells which do not have nucleus. Now, where is this nucleus found? Because it has the function of controlling the cell metabolism and other cellular activities. Mostly, you will find it in the center of the cell. Okay, it will be mostly towards the center of a cell. In plants, it gets pushed to one side. Okay, in plants, you have this huge vacuole, and because of this vacuole, this is your nucleus, which gets pushed to any one side. Then, number of nucleus. Commonly, all the normal cells are uninucleate or mononucleate. Uninucleate or mononucleate means having one nucleus. Okay, these cells are called uninucleate type. So they have these single nucleus present inside. Then you have some organism which are binucleate, which means they have two nucleus or two nuclei, plural. Example is paramecium. Here, this is a paramecium where you can find there is two types of nucleus. One is macronucleus. Macro means larger in size. And then you have a smaller nucleus called micronucleus. Here, the function of two nucleus are different. The micronucleus will be used for giving it to the daughter cells, okay, for genetic inheritable properties. While the macronucleus, it has the function of controlling the cellular activities. Fine, so that is how they are divided. Multinucleate condition is found in some cells. In some cells, you find more than two nuclei. So this is called a multinucleate cell or a multinucleate condition. In case of animal cells, these are called syncytial cell. Okay, for example, the cell of your bone marrow or striated muscle, skeletal muscle, one and the same. Fine, voluntary muscle. So this is your one muscle cell, which is called a muscle fiber. Fine. So this one muscle cell or muscle fiber, you can see has a lot of nucleus. Okay. So there are multiple nuclei present in this animal cell, and this is called syncytial cell. In case of plants and fungi, These kind of arrangement which has a lot of nuclei is called choanocytic cell. Okay, it has many nuclei. As you can see here, this is just one cell, okay, which is having a lot of nuclei. One thing I would like to say is the both conditions are arising by fusion of single cells. Okay, you have cells called myocytes. These myocytes, when they fuse, okay, they form this single cell like structure which is called muscle fiber. Fine. Here, in case of fungi, it is known as a single mycelium. So initially these are not single cells, but they are fusion of different cells. Afterwards, because there is no division in between, you can call them a single cell which has multiple nuclei. Fine. Shape of the nucleus, they are normally rounded in outline, so they are round or spherical in shape, and they are disc shaped in squamous epithelium. Squamous epithelium is where the cells are flat and square in shape, okay, like tiles on the floor. So in such kind of epithelial cells you have disc shaped nucleus, while they are lobed in WBC. So just focus on this diagram. Let us go through them one by one. Now here, as we have already discussed, in paramecium they have two nucleus, a macronucleus, a micronucleus. Then this is your RBC, which does not have any nucleus. These are the different kinds of WBCs. Okay, you have studied them, right? The monocytes, and then you have granular leukocytes. So in different kinds of WBCs, you have different kind of or differently lobed nucleus. Fine. Lobe means these rounded structures. Okay, these parts, subparts of a whole structure. Fine. So here you can see it in horseshoe shape, in kidney bean type, then kidney bean type shape. So in case of WBC, they can be multi-lobed. Fine. Then in some cases, in some alga, they may even be bead shaped like this, or they in a polynucleate cell, which means it has more than one nuclei. You can find it like this. Okay, so you have different shapes and sizes of nucleus. Now let us discuss the ultrastructure of nucleus. A typical interphase nucleus. Interphase is when the cell is resting. There is no cell division going on. The cell is at rest. In such cases, the nucleus is 5 to 25 micron in diameter. Again, this range is given because depending on the cell type, the organism type, the size differs greatly. The nucleus is divided into 5 parts. The outermost nuclear envelope, okay, the covering, the nucleoplasm. It is like how cytoplasm is the what you can say watery part of a cell. Same way, the watery part or the liquid part of your nucleus is called nucleoplasm. Then the cytoplasm has cytoskeleton, right? The same way your nucleus, to maintain its shape and functionality, has nuclear matrix. Fine. The nucleolus and chromatin. Let us start with the nuclear envelope. Here it is made up of two membranes which are separated by a perinuclear space. You can see here this is the outermost membrane, and then you have an innermost membrane, and there will be spaces in between. That also you can see. These spaces are actually nuclear pores. Okay, they are holes, they are pores in the nuclear envelope. The outer membrane is often connected with the rough endoplasmic reticulum. You can see this is the outer membrane, and that outer membrane is connected to the rough endoplasmic reticulum. Clear? One more thing I wanted to clear, why it is better to call it nuclear envelope and not nuclear membrane is because this membrane is not continuous. Okay, it is not continuous, rather it is present in form of some pockets. Okay, so here for example, if you see, then this will be the outer membrane which will take a U-turn and form the inner membrane, okay, which will again take a U-turn and be connected on all sides. Fine, and there will be pores in between. So it is better to call it an envelope rather than a membrane. A membrane is the one which is present throughout, okay, equally. While here you can see there are a lot of gaps in between. So it is better to call it nuclear envelope. Because it is attached to the outer membrane, the outer membrane is attached to the endoplasmic reticulum. Sometimes, very rarely, you find ribosomes present on the outer membrane of nucleus, okay, not on the inner membrane, on the outer membrane. But these these ribosomes, they do not have any function like synthesizing protein inside the nucleus, okay, nothing like that. They are just attached on the outer membrane because that outer membrane is continuous with the endoplasmic reticulum. So sometimes it is difficult to say whether it is the part of your outer membrane or it is the part of your endoplasmic reticulum. For example, this site that you can see. Okay. Nuclear envelope consists of a large number of pores. You can see a lot of pores are present. They are made up of proteins called nucleoporins. Okay, this is the name of the protein which makes these kind of pores. These pores, they allow transport of RNA and proteins in and out of the nucleus. Okay, so whatever mRNA, tRNA, rRNA are synthesized in the nucleus, they will be leaving the nucleus and entering the cytoplasm through these nuclear pores. However, it will not allow DNA to leak out. Okay, it will still keep the DNA inside it. The nuclear pores control passage of substance like RNA, ribosomes, and proteins. After nuclear envelope, let us discuss the nucleoplasm. As I already told you, like cytoplasm for the cell, nucleoplasm for nucleus. It is transparent, semi-fluid, colloidal substance which fills the nucleus. This liquid portion has a lot of DNTP. DNTP is deoxy ribonucleotides. Okay, DNTPs are used to make your DNA. Then it has a lot of NTPs, normal ribonucleotides. Okay, uh, these ribonucleotides are the ones which were monomers. Okay, and they are used to make the different RNA, mRNA, tRNA, rRNA. Nucleoplasm also has certain enzymes and proteins. Enzymes include the different polymerases like DNA polymerase, RNA polymerase, etc. are all present in the nucleoplasm. Clear? Then you have nuclear matrix. Nuclear matrix is a network of intermediate filaments. So if you look at this diagram, the bottommost diagram clearly, you can see this is the nuclear pore which is made up of nucleoporin. Okay, and beneath that nucleopore you can also see these blue kind of fibers and these greenish colored fibers. These fibers are nothing but they are intermediate filaments. Okay, and they provide structure and support to the nucleus. It forms a dense fibrous layer beneath the nuclear envelope. So this is your nuclear envelope, and beneath the nuclear envelope you find this thick and dense band. Okay, and this band is called nuclear lamina. Fine, nuclear lamina. It is made up of lamin protein. Okay, so this is all about the nuclear matrix. The functions, of course, include scaffold for chromatin, the red colored substance that you see. Okay, that is your DNA. Now this DNA will not just be roaming around and floating around in the nucleus. It will be at some points attached to this nuclear lamina. Okay, to provide this what you can say support. Okay, and this nuclear lamina or the nuclear matrix is present. Then it gives mechanical strength to the nuclear envelope. It gives shape and strength to the nucleus. Right. So this is the microscopic diagram, okay, photograph actually of the nucleus. You can see the darker portion is called nucleolus. Then you have the outside portion which is nuclear envelope, And inside the nuclear envelope, the thick dark colored band you see is your nuclear lamina. When you look at the surface of this nucleus, you find it like this, okay, dotted, and each of these dots are nothing but they are the nuclear pores. Now we move on to the next part, fourth part, which is chromatin. Now, as we already know, nucleus is the organelle which consists of all the DNA. This DNA is not present alone. Okay, it is present in form of a complex with protein. So it is hereditary DNA RNA DNA protein complex. Okay, it has some RNA also, but here we'll just focus on DNA and protein complex. Okay, so this DNA protein complex is known as chromatin. It is called chromatin because chroma, as you already know, means color. So this portion of the cell, okay, of the nucleus, it gets dyed easily with help of basic dyes. Why basic dyes? So if you know, DNA is negatively charged because of phosphate groups, and this negatively charged DNA is acidic in nature. So if you want to stain something which is acidic, you have to use basic dye. Okay, so if you use any basic dye, it will be able to stain this part of your cell, and that part is nothing else but chromatin. Chromatin is a complex of protein and your nucleic acid, which is DNA. The proteins here are histone proteins. Okay, the protein mentioned in this protein complex are histone proteins, and these proteins are rich in basic amino acids. When we will discuss amino acids, we will see that there are different kinds, okay, different varieties. Some of them are basic in nature. So example being arginine and lysine. So histone proteins are rich in arginine and lysine. Because they are basic in nature, the acidic DNA can easily be present around it. Okay, so we will see that histone proteins are like round spheres, okay, like ball like structures, and on these ball like structures you have to wind the DNA. Okay, so this winding of DNA or arrangement of DNA is only possible if there is some attraction between the histone and the DNA. DNA is negatively charged, it has acidic nature, and the histone is having basic nature. So this is how you will be able to properly bind the DNA to the protein. Chromatin is fibrous, okay, it is fiber-like, and it takes part in DNA replication, as well as it controls the cell metabolism. Chromatin is seen in resting cell, that is in interphase nucleus. When the cell is not dividing, it is at rest, you find in the nucleus presence of chromatin. They are differentiated into two regions. When you stain it and when you look at it, you can find the chromatin is differentiated in two regions. The region which is lighter in color is called euchromatin, and the region which is darker in color, as you can see, okay, it is patchy. So the darker in color is more dense, and it is called heterochromatin. Clear? Here these are the heterochromatins, and the white colored portion is your euchromatin. Now let us study the chromatin condensation. To condense anything is to bring something together, okay, to fix everything in small space. Now your DNA, if you know, the human DNA can be as long as 2 meters. So this 2-meter long DNA, how to pack it in a small space, okay, your nucleus is only 25 micron big. So how to fix this long 2-meter DNA into a small nucleus? This is done by condensing the DNA, okay, and this is called chromatin condensation. Let us look at it stepwise. Okay, here you have your normal DNA, which is 2 meters long, and it has diameter or width of 2 nanometer. This is your normal DNA in form of thread. Now what you do is, as I already told you, there are proteins called histone proteins, which are showed here in blue color. These histone proteins, on these histone proteins, the DNA gets wound. Okay, so the DNA, it makes 1.5 turn on each of these histones, and this is how you reduce the length of the DNA. Let us see how. Okay, so the histone protein is not a single protein. It is made up of 4 different type of proteins found in copies. Okay, so it is a histone octamer. Octa means 8, because there are 8 proteins. Here you can see 1, 2, 3, 4, 5, 6, 7, 8. Okay, there are 8 proteins together. There are 2 of each type. How many types are there? 4 types. There is a type called H2A, H2B histone, H3 and H4. Okay, and behind this H3 there will be one more H3. Behind H2A there will be one more H2A. Right, so this is how there is an octamer. There are 8 proteins, 8 histone proteins coming together to form one complex. Okay, so H2A, H2B, H3 and H4. On this, the yellow colored portion that you see is your DNA. Okay, so the DNA is wound on this histone, right, and it forms this nucleosome. It is called nucleosome. Clear? There is also one more histone protein called H1. This H1 histone protein is found single. It is not found in any dimer form. Okay, it is single, and it is also not attached to this complex. It is found in the linker DNA. Fine, just look at this diagram, bottom diagram. The blue colored ones are your histone complexes, the octamers, and the red colored one is nothing but your DNA. So your DNA is coming like this, it is winding around the histone and then leaving and then winding around another histone. So there is some distance, there is some free DNA between the two histone complexes. This is called linker DNA, okay, because it is linking to nucleosomes, and this linker DNA has H1 histone. Clear? Fine. Now these um, structures, they will come together and they will form one more round. Okay, they will wind up like this. This is called your solenoid like structure. Fine, it is 30 nanometer in width now, but when you look at it from above, or you look at the cross section, you see it like this. There are 6 to 7 such complexes arranged in a flower like pattern. Okay, this is how they will get arranged. Can you see, little by little we are reducing the length of this DNA. Okay, we are winding it over and over, and this is how we are reducing the length of the DNA. Fine. After that comes this portion where you can see some other kind of protein. Okay, this yellow colored one is some other protein, and this thick fiber like structure you are seeing is nothing but this solenoid. Okay, this, consider it as one wire. Okay, and this thick wire of 30 nanometer is wound in such manner. Okay, such helical type shape. This helical shape is maintained by some proteins. Okay, so these are some other scaffolding proteins. For example, they could be condensin, right, that you find here in the chromosome. Can you see this middle portion on which more and more DNA can be arranged? Now these thick, again, fibers are considered as one unit, and they are snaking around like this. Okay, and they form one more huge flower like structure, and this actually is a part of your chromosome. Okay, if you take this much part of your chromosome and you look at it, it will look like this. Fine. So this is how the DNA is condensed. Okay, it is reduced in length, it is wound over different proteins to make sure it is less in length. Okay, and it increases in the thickness. So this is the nucleo protein complex. Okay, and this is how it is forming. There are different proteins involved. The first and foremost proteins are called histones, and later on there are other proteins as well, like condensin, which help in production of chromosomes. So this is how a single strand of DNA, which can be 2 meters long, is converted into a chromosome, which may be smaller in size, like 1 micron. Okay. Now let us consider the differences between euchromatin and heterochromatin. Remember in the diagram we just saw, okay, one slide ago. So euchromatin, it is narrower, okay, it is thin. It is mostly a 10 nanometer fiber, which is called beads on string, because it looks like that, okay. It looks like there is a string and there are beads on it. So this is called bead on string, and the 30 nanometer fiber is called solenoid. So 10 to 30 nanometer fiber, euchromatin is made up of such fibers, okay. It is narrower, while heterochromatin is made up of fiber which is more than 100 nanometer thick. Euchromatin is diffused in the nucleus and it is lightly stained. It will mostly appear clear, okay, in color. While heterochromatin is condensed. It is much more dense and it is darkly stained. Nucleosome strand has minimum coiling here, while the nucleosome strand has solenoid or more than solenoid kind of coiling in heterochromatin. Fine. Euchromatin is fibrous while heterochromatin is granular. Euchromatin has active genes because these DNA strands, they are a little open, okay, they are not wound so much. All the genes are active, they can be easily transcribed, while this heterochromatin is so tightly wound, it is so thick and dense that you do not see transcription activity in heterochromatin. Then this euchromatin is affected by pH, temperature and hormones, while heterochromatin is not influenced by any of these factors. The euchromatin, during cell division, it replicates early, while the heterochromatin, it gets replicated late in the synthesis phase, okay, S phase of the cell division. Here in the diagram you can see this is your euchromatin, which is very diffused, okay. It does have the nucleosome, but they are comparatively diffused, while in case of heterochromatin they are packed densely, okay. There is less amount of free DNA. Right, we now come to the last part of nucleus, which is nucleolus. Okay, we discussed 5 different parts of nucleus, and this is the last part on the 5th part. Nucleolus is also naked. It is not any different structure with membrane or anything. Okay, it does not have a membrane, which means it is naked. It is round or slightly irregular in shape and structure. It has some specific regions, okay. These yellow colored portions you are seeing are your nucleolus, sorry, okay, nuclei. So here the yellow colored portion, when you zoom in on one nucleolus, it looks like this. Here you have the centermost portion, which is called nucleolar organizer region, okay, NOR. This nucleolar organizer region is a part of chromatin, okay, it is part of your DNA, and these chromatin, they form loops like this in the NOR region, okay, nucleolar organizer region. These regions actually contain the DNA for synthesis of your rRNA, okay. Your ribosomal RNA are encoded by proteins and by genes, my mistake. So these genes, okay, on your chromatin are present here in the nucleolar organizer region. Fine. So if somebody asks you what is present in NOR, NOR has the DNA, the chromatin. This DNA has the gene for making rRNA. Then you have the next region, which is called fibrillar region, okay, because it looks fibrous when you look at it under the microscope. So this fibrillar region has a lot of rRNA which is getting transcribed from the DNA. Fine. So this dense fibrillar region has nothing but your rRNA getting transcribed. As you can see here, these purple colored whatever strands you are seeing are your different kinds or different length of RNA. Fine. And finally, you have the outermost cover, okay, or the outermost region, which is granular component. It is granular because it looks like it has a lot of dots, okay, it looks granular. The reason for this is there is presence of protein complexes, okay, there are different proteins which have come together. These proteins are nothing but they are your larger subunit and the small subunit of ribosomes, okay. So the pre-ribosome assembly, that is your large subunit, your small subunit, your rRNAs, they are all present in this region, okay. And so this is called the granular component of your nucleolus. Commonly, in a human nucleus, there are 1 to 4 nucleoli. Okay, in one nucleus, there are 1 to 4 nucleoli. Now, there are these 5 chromosomes 13, 14, 15, 21, and 22, which have this NOR, which is the nucleolar organizer region, which means it has DNA to produce rRNA. Fine. So, maximum you can have are 5 nucleoli, and the minimum you can have is 1 nucleoli. Clear? Now we discuss what these chromosomes are. I hope till now it is all clear to you. Chromosomes, as we discussed during chromatin condensation, so they are nothing but they are condensed form of protein plus DNA complex. Okay. Each cell has a pair of each kind of chromosome. Each kind in the sense there are some number of chromosomes, like in humans there are 23 pair, right? So each cell has a pair of each kind of chromosome, okay, from 1 to 23. They are known as homologous chromosome. These homologous chromosome, the pair, one you get from your mother and one you get from your father. Chromosomes are made up of chromatin plus other protein. Okay, chromatin is your histone plus DNA, and apart from histone plus DNA, you also have some other proteins like condensin present in the chromosomes. Each chromosome consists of thousands of genes. So if you take this as a one chromosome, it will have many genes. Structure of chromosome can be best observed during the M phase, okay, metaphase of M phase, mitosis phase, okay. You will find the chromosomes to be condensed maximum during metaphase, okay. So here is when you can see these chromosomes very clearly in the cell. What is its function? Why is a chromosome required? It is for equal distribution of genetic information to the daughter cells. Fine. So in a normal cell which is not dividing, this is the kind of chromosome you find. Okay, mostly it is in form of chromatin. But if you forcefully ask the chromatin to condense in lab, then it will condense in form of this structure. Okay, it does not have two arms or two legs as you have always imagined the chromosome to be. Okay, chromosome in normal cell has this kind of structure. Okay, the smaller arm or the upper arm is called P arm and the lower arm is called Q arm. Clear? Both of these arms are connected by a centromere, and then you have the ends which are called telomeres. In a metaphase chromosome, that is after multiplication or after replication of your DNA, now you have two copies of the same DNA, which is why the P arm will also have divided in two. You have two P arms, and Q arm will also have replicated in two. So now you have two Q arms. These are called sister chromatids because they are identical. They are same. Okay, they are the same DNA. And all of these will be connected through the centromere. So these are the parts of the chromatid, chromosomes. Okay, chromatid, it contains a single DNA molecule, which means if somebody asks you how many DNA molecules are present in a human cell, The answer would be 23 pair or 46 in total. The metaphase chromosome have symmetrical structure called sister chromatids. Okay, these are the sister chromatids. At anaphase, where you have to separate these sister chromatids, they will get separated through this middle, okay, and that middle portion through the centromere, the splitting will take place. Centromere is where the two sister chromatids are joined. Then the spindle fibers during the cell division, okay, that you see here, will attach at the centromere. Specifically, if somebody asks where in the centromere, there is a small region, a disc-like structure called kinetochore. Okay, this kinetochore of centromere is the place where the spindle fibers can attach. This kinetochore is present on both sides of a single bivalent or a single chromosome. Okay, so this here is your single chromosome, and on that chromosome on both sides you have a kinetochore. Then telomere as is the last, okay, the tail portion of a chromosome. It is the terminal part of the chromosome. Then satellite is like this. It is an elongated segment. It is sometimes present on a chromosome, and such chromosomes are called sat chromosomes. Fine. And there is a secondary constriction meaning there is some narrowing, and after that it will be bulging out. Okay, so these are the satellites of the chromosome. Very rarely you find these, okay, only on some chromosomes. At prophase, the chromosomal material is visible as thin filaments. Okay, so the chromosome at prophase would have condensed a little and it looks like thin filaments. Okay, so these thin filaments are called chromonymata. At interphase, bead-like structures are visible, which are accumulation of chromosome or chromatin material called chromomere. So the chromatin we had studied, they come together and form some granular-like structure. These granular structures which are visible during interphase are called chromomere. Some of these terms, they are not very much important, but just in case if they do get asked, you should know them, okay, at the back of your mind, or you should have at least heard of them sometime. Fine, so that is why I've just mentioned these terms. They are very rarely asked in your exams, but you should know just in case if it gets asked, or if there is a question which is mentioning such name, then at the back of your mind, if you have a little idea, you may be able to answer. Now we move on to the types of chromosomes. There are different types based on the appearance, okay, or the presence of centromere, where it is present on its location. So the first one is called metacentric. Metacentric is V-shaped, as you can see here. Fine. Though you draw it like this, you when you look at it under a microscope, it is mostly like this, bent a little. Okay, and because the centromere will be present exactly in between, it will give rise to two equal arms. Fine. So this is called metacentric, which means the centromere is exactly in the middle. Then you have submetacentric, which means the centromere is present a little away from the center, okay, on any one of the side, which gives you an L shape. Okay, when it will be bent, it will give you an L shape or a J shape. So these are submetacentric, where you have one arm which is a little smaller and another arm which will be a little bigger. So unequal arms. Then you have acrocentric, where the smaller arm, okay, you have one very short arm and one very long arm. So the centromere is present almost towards the end of the chromosome. And finally, you have telocentric, where the centromere is present exactly on the tip and you just seem to have one arm. So this here is your telocentric chromosome. Telomeric chromosomes are not found in humans. You do not find such chromosomes, telomeric chromosomes, in humans. This is just an example of the chromosome numbers, okay, in different organisms. The 2n means the diploid condition. Okay, diploid condition is where you have the homologous chromosomes, one from your mother, one from your father. Then you have haploid conditions seen in the gametes, okay, in the sperms and ova. So these cells will have haploid, that is only half number of chromosomes. So this is called N condition. Okay, haploid is N, diploid is 2N. So in Ascaris, which is a roundworm, okay, Escalminthus, it has two chromosomes only, and in haploid cell it will have only one chromosome. Then Drosophila is fruit fly. It has 8 chromosomes, and in its gametes there will be 4 chromosomes. Chimpanzees or gorilla, which are our closest relatives, they have 48 chromosomes, and we have 46 chromosomes. Fine. There is a protozoa called Olocantha. This Olocantha has 1600 chromosomes. Fine, so you can see how much there is difference in the number of chromosomes. Okay, it could be various. Same thing you can also go through this table for different plants. Okay, these values again are not important to remember each and every one of them. Just remember few of them. Okay, remember Drosophila, it has 8. Remember onion also has 16. Okay, so half will be 8. Then chimpanzee or gorilla has 48, which is closer to our, which is 46. When you stop a cell at metaphase, okay, you arrest a cell at metaphase and you take a picture, you will find such kind of a photograph where you have different chromosomes arranged like this inside a cell. So what the scientists do is they look at the banding pattern. Can you see there are different bands seen? Okay, so depending on the length and the banding pattern, they pair up these chromosomes. Okay, so this here is chromosome number 1, and this is its pair. We said in the beginning, right, that the chromosomes, they occur in homologous pairs. So here one will be from your mother, one will be from your father. Then chromosome number 2, one from each parent, 3, 4, 5. And similarly, you have as many as 23 pairs. Clear? So this is called karyotyping. That is, you are arranging the different chromosomes from your cell. Okay, the photograph that you have taken of your cell. From that, you are arranging the chromosomes in pairs and in order. There are two special chromosomes which I would just like to mention briefly. There are some chromosomes called polytene chromosomes. Okay, these polytene chromosomes were found in the salivary gland of Drosophila. I'll tell you what Drosophila is. Have you ever left bananas on table? Okay, and if you see on those bananas after some days, you will find some red or brownish colored small flies which will be flying around. Okay, even on the garbage, very small flies which are reddish in color. So these small flies are called fruit flies or Drosophila. They feed on fruit, okay, which is why you will find them mostly around fruits and vegetables. Fine. So in the salivary glands of these flies called Drosophila, such chromosome was found. Okay, this is a microscopic image. You can see this is one single chromosome. It took scientists a long time to understand that this was just one chromosome. Okay, you find it in the salivary gland of Drosophila. You also find it in larval stage of Diptera. Okay, Diptera are all the flies. It includes house flies, and it includes all the different kinds of flies there are in the world. So in the larval stage of such flies, you find polytene chromosome. Here the DNA will multiply multiple times. Okay, it will undergo multiple rounds of replication, but there will be no division, which means all of these sister chromatids, they stay attached. Okay, and this is how a polytene chromosome is made. And because there has been multiple rounds of replication, the DNA is very large. Okay, the chromosome is also very large. For example, Drosophila's polytene chromosome has about 1000 DNA molecules. Clear? That is how big it is. When you look at it, you find there are some stripes. Can you see? There are dark bands and then there are light bands. So this alternating dark and clear bands give it a striped look. The clear or the lighter bands are also called interband. Okay, that is the region between two dark bands. Then at some area you will also find there is swelling. Okay, there is puffing. So this portion is called chromosome puff because it is swollen like that. It is rounded like that, and it is also named as Balbiani ring, on in the name of scientist who actually observed this structure first. So the scientist was Balbiani, who observed this polytene chromosome first, and so this puff is named on his name. Okay, Balbiani rings or chromosomal puff. These are swelling of the bands because there is loose arrangement of DNA. Okay, the DNA has unfolded. There is less condensation in this portion. Because there is less condensation, you can easily transcribe here. So wherever there is high transcription activity, that is a lot of mRNA is made, you will find such sort of puffs. Okay, here the genes are active and they are easily getting transcribed into RNA. The second kind of chromosomes, which I have forgotten to convert the name, it is actually called lamp brush chromosome. Okay, this is not polytene chromosome, this is lamp brush chromosome, pardon my mistake. So it was first discovered in oocytes of salamander. Salamander is sort of like a lizard. Okay, but it is of group amphibian. So in salamanders it was first discovered in the oocytes, which means the egg cells. They occur in diplotin stage in many oocytes and spermatocytes of animals and in algae like acetabularia. Remember, we have studied acetabularia when we were discussing the shape and size, okay, of different cells in very beginning. They are present as bivalent with 4 chromatids. Okay, they have 4 chromatids. The lateral loops, okay, fine. Let me just describe how they look like using this diagram. So here you have these 2 chromosomes, one from your mother, one from father. Okay, so maternal chromosome, which means from mother, and paternal chromosome, and they are connected in this way. So they form a bivalent structure. Now, when you look at this cross section, okay, you can see this. There is something coming out of it, right? So when you zoom in on that portion, you will find that the DNA, it is dense in the center, in the axis. Okay, so here in the axis, the DNA would be found condensed. Okay, and that portion, that condensed portion, is called chromomere. Okay, granular portion. Remember. And from this portion there will be equal length of loops coming out. These are nothing but these are DNA. Okay, DNA will be looping out, and these loops are equal in length. Fine. So these loops are the parts or the points where there will be presence of genes, and from these loops or from these genes the RNA will be made, mRNA. Fine. So it is symmetrical, that is same on both sides, and from these places the mRNA is made. In oocytes of salamander there are 10,000 loops present per set of haploid chromosome. Okay, so if you take a haploid chromosome, which means it only has any of the one, either from mother or the father chromosome, and you look at it, you will find 10,000 loops, okay, present on one set of chromosome, haploid chromosome. Fine. This is how it looks like, the chromomere, which is a condensed form of the DNA. Okay, it looks a little granular, it looks a little dark, and when you look at the loop, it is a single strand of DNA going out from the normal axis. Clear? So here we end the chapter cell. If you have any doubts, please reach out to me. Otherwise, the next we will be starting with the cell division and cell cycle.